Hello everyone and welcome to the very first lecture of MSAI computer vision course, and today we will discuss biological vision systems. This is a very important topic since all of us have such a system from the start, from the birth, and in fact evolution have made really beautiful systems of vision which we could not reach with our artificial technical stuff uh, till today. I guess we will be able to do that in future, but I guess for maybe the next decade, I don't think that we will beat the benchmarks by biological systems. So let's discuss the outline, and first I will speak about some course information, general information, then eye structure, especially in human, some words in binocular vision, and we will end up with the application in science and business. This is what we all want to do. The first thing is a course info and a couple of words of myself. My name is Vladislav Goncharenko. Currently, I'm a head of perception at Evocargo. I do some mathematical models such as neural networks and others, classical models, which first takes data from sensors, then processes it, and with some classical stuff such as stereo vision, such as distortion fixing, and so on. And finally, we use neural networks for detection, segmentation, and so on, depth estimation, many problems. Then I'm doing my PhD work at IITP in laboratory of vision systems, and I will tell a couple of words on that laboratory during my presentation. By the way, I create and taught courses on machine learning, on computer vision, different topics in different places. And time after time, I try to contribute to open source projects. You could check out my GitHub, you could check out my LinkedIn if you want, and with all the questions, you could reach me in Telegram. So the next part is a course plan, and it is divided into two parts. We kind of started the first part with the webinar, and we also kind of started the second part with a lecture and seminar. The first part is classical computer vision, and nowadays many computer vision specialists, especially deep learning specialists, don't know much about what happened before the neural networks came. And people tend to think, for example, that convolutions were not did not existed before the deep learning, and so on and so on. It is not so, and we will go through it from image forming model, like a physical model. We will go to representation computer and why it is done in this way and not the other. We'll go through color theory and many techniques of image processing. We will end up with the kind of pre-neural networks, for example, histograms of gradients. That is what used before neural network segmentation for solving task of image semantic segmentation. And we'll smoothly go to the second part of neural computer vision. Here we will bring some light to CNNs, which I think you are familiar with, and go deeper into different tasks of computer vision, such as image classification, object detection, semantic segmentation, and learn a bit about style transfer. We'll tell a couple of words on transformers in computer vision, which nowadays came to this sphere and definitely came to stay, and so on. In fact, many different topics, just uh, just a part of it are presented on the slide. But two parts are distinctly exist, and let's move on. Today's topic is biological vision systems, and we start from a human eye. I guess all of you have seen such an image in your textbooks on school biology and so on. And from this picture, you can see that human eye is a very complex structure which consists of many different parts, each of which is responsible for something different and is important for all the systems, for all the system to work properly. Many muscles, many types of tissues, and so on. To motivate such a complexity, I could say that later, at the end of summer, I I ride my bike by the street, and the sun shined straight to my eye, and still I was able to see people which walked around me, some obstacles, and could successfully drive my bike. And I thought to myself that modern camera, even the best ones, could not handle such a complex situation of being overlighted. And yeah, I think that maybe in decade or something like that, we won't be able to beat such a highly placed level of perception system. But still, I think we could take one and other ideas from biological systems and move it to our technical vision. And step by step, we will, I think, reach out the competitive level of quality. Let's bring it to our knowledge. And now let's go deeper into biological vision, and let's start from evolutionary development of vision systems. First, on the surface uh, of alive forms of existence, we had uh, we had some nerve fibers which went to the surface, and there were some photoreceptors and special cells for that, but they were just part of the surface of an kind of animal, but I think not animal, but some less complex form of life. And step by step, we developed the system that consisted in a chamber that light could go into. And later in this chamber, we had formed some refractive lens, and the nerves get organized better and better. And step by step, we went to iris and cornea. This is more and more advanced types of eyes. We will see more on it on the next slide. And this is how it formed. So this is a human-like chamber eye, and in fact, all the eyes of animals could be divided into two big groups. First one is chambered eyes, which I already described a bit, and the second one is compound eyes, which is common for insects and for different forms of life. This is the scheme that demonstrates the structure, and we'll mostly speak about chambered eyes because compound eyes, in fact, mm, is used less in technical vision, uh, and it consists in many sensors, which each of which is less complex than one eye, but it provides different characteristics of vision. For example, angles could be better, and we could see more detailed from kind of detailed, but in some different notation information, and we will not speak about it more. You could see more eyes classification by these links, but by this link below, and we will stop on differences of chambered eyes. Here we have two types of chambered eyes. The first one is from octopus, and here in common you see the lens, some muscles. You see, for example, sclera. This gray is sclera, and optic nerve. This is common parts. But what differs here is that photoreceptor cells, in the first case of octopus, lays on optic on nerve fibers, which lays on the sclera. And this is common for animals which live underwater. And another type is photo of an eye, of a chambered eye, is that photoreceptor cells lays directly on the sclera, and nerves go from outside of this, from inside of the chamber, and it guarantees better stability of photoreceptor cells than in case of octopus. And this nerve fibers is more movable than sclera. And in fact, when photoreceptor lays directly on sclera, it is fixed, and we could achieve better stability and more detailed picture. This is one thing, and another difference is that in the second type of eyes we have blind spot, which is created to for optic nerve to be able to go out the chamber. In this case of a human eye, we have a layer of some fibers preventing our eye from overlighting, because when an animal lives underwater, it receives less light. The light underwater is more distracted, less focused when it reaches the eye, and that's why the case of overlighting is not common for underwater animals, and for those who live on the surface of Earth, it is not so, because we could see even direct beams of light, and this is the reason of existence of bipolar cells, which besides that they provide signal from photoreceptors to nerve fibers, they also prevent our eyes from being damaged from direct light. This is the differences in these types of eyes. As I said, you could check out the link for more information, and we will go on. We have different types of cells for perception, and first one is ganglion cells, then bipolar cells, and this is an nerve fiber which goes to optical nerve, then bipolar cells, which is kind of medium, and the photoreceptor cells are mostly rods and cones in human eye, and I guess you have heard about it. This is amplitudes of light, uh, which triggers rods and cones. And cones is made to see in evening time and morning time, at dawn, and rods is for colored vision. uh, so okay, uh, then I wanted to add that many of research on eye functioning were made at IITP, which I mentioned before. This is the institute that I'm studying currently. And here is the book from Professor Yarbus on eye movements and vision, written in 1967, which in fact was a table book for vision researchers. Many information that is collected in vision laboratory of IITP and presented by Professor Yarbus. For example, here we have distributions of cones, spectral response activities, and this is uh, the system for eye tracking from 1967. Nowadays, a couple of slides later, I will give some ideas how these systems looks like nowadays, but many of these research were made at IITP. You could check out the book and maybe read it, because most of the things is still presented, and they were digged deeper and developed, but still the basis is here. And on this slide you see retina spatial sensitivity. On the left we have the bottom of our eye, of our chamber, and it is a retina, it's called retina. And on the right we have distribution of density of photoreceptor cells sensitivity, starting from fovea. And the fovea is placed on our retina with the highest density of such cells. Usually all the measures are made from this point, and this is a zero angle. It is this phenomenon is caused by the high cost of such cells, because they are hard to produce by our organism. And here we see that the maximum density is in fovea, and in everything, in every other place we don't have in fact colored vision, but rods are distributed more widely and kind of more uniformly, and with rods we can see just some movements and so on, and we can see at dawn with rods, and both of cones and rods have zeros on the blind spot, which is about 12 degrees from the fovea. And this is how our human eye is functioning with these types of cells. This is more detailed representation of our spectral sensitivity, and here we see that different cones is created for different types of light. Here we see blue, green, and mostly red light. This is three types of cones that we have, and we have only one types of roads, which is about 498 nanometers, and this is from violet to red scale of wavelengths. And on the right you can see different bottoms of a chamber, like retina, of different species. For example, zebrafish, chicken, and so on. And on the bottom you can see human, and by the by increasing of complexity of an animal vision system, we can see more and more concentration on the center of the field, uh, and different distribution between an eye bottom. And for human we have this fovea, and speaking about these types of our cells, we'll speak more about it in the next lecture, when we will try to describe the picture with some computer facilities, represent picture in computer memory. And this is how we will do that. We will have 3 channels for each type of cone that we have in our eye. Speaking about angles that we see, it is measured from the gaze direction, and all uh, the fovea has about 7 megapixels, and it is only 5 to 10 degrees, or even less. In fact, we have this foveal space about 5 degrees, and if you want to maybe estimate your fovea, you could place your hand like this and look at the tip of your finger on your nail. And if you look on your nail, this is the angle that your fovea has. So all the other angles we see less less distinctly. For example, we could see shapes for 5 to 30, colors only to 30 to 60, and motion, this is the most important for living in the wild world. We see for 84 degrees. Also, signals from our optical system is processed in different with different speed, and motion is the most rapid thing, which is processed with the lesser value, and more detailed information goes up to 500 milliseconds. And let's imagine how our vision of one eye could look like if we measure it. If we receive one instant signal from an optic nerve. This is the reference picture. This is what we see with the camera, for example. This is just a photo. And let's imagine what we could see with just one eye. And this is what we have. First, it's flipped upside down because we have the lens, and the lens have the property to move, to kind of shake everything upside down due to physical things. We will speak more about it on the next lecture. Here we have a blind spot and vessels that stops our light from being perceived. Then we have only motion on the corners of our image, some shapes more centered. Color vision is only about 5 to 10 degrees, and in fact really sharp image exists only in the center of this shape, because and we have a fovea region only in the center. And even if we add the second eye, we will see more the angle increased, but still color vision is only in center, and foveal region is just tiny thing. And this is how we really look, what we really see with our eyes. All the other picture came from processing it by our brain, because our vision system could provide just this picture at a time. Uh, and if you drive the car, I think you have heard about this phenomenon of blind spot, and that if you want to make some movement, you have to look on right, look on left by moving your head. This is caused by the blind spot existence, because we could just not see some other automobile, for example. And let's think what happens next. So I mean, we in our brain we have different image. We have image like this when we look at any direction, yeah. And how it forms is some magic, and all of the technical vision systems and nowadays kind of artificial intelligence systems try to do the same things with our technical stuff. So this is another picture how our vision degrades depending on the angle of our motion. You could stop the video and read everything here. This is very interesting, and yeah, this is how our vision operates. So let's go to the processing of the picture from our eyes. And research shows that information by an optic nerve go to the rear part of our brain, to foveal region in our brain, and all the vision happens at the rear part. And in fact, right and left eye are processed in this way. This is like common picture from textbooks on brain biology. Yeah, foveal field is formed here and processed by two parts, and then when signals are processed in each part, they create a more complex picture of binocular vision. And let's speak more about it. Binocular vision, here you have some fields of view depending on where we look, of our gaze direction, and this is the fields, and this is how our binocular neurons operates on different parts of this field. And on the right you can see angles for different animals. And the fact is that binocular vision here is very useful for predators. They usually use binocular vision for hunting, because they have to go to their victim. And so the victims of predators usually have less binocular vision field, but more just monocular vision and less blind area, because they are hunted and they want to know where the hunter came from. It just go to opposite direction, like we do in gradient descent, for example. But they do it for their life and to surrender. So this is how binocular vision operates in the case of a human, and this phenomenon is connected with stereo vision, and it is called autostereogram. You could stop the video and look kind of through the screen of your monitors, and you will see the shape. In fact, there are many articles on that and maybe popular information, but this is connected with how our eye processes the information from our binocular system. We could see some image here, not contained, not connected with some microprocessors and so on. When we have a binocular system, in fact, as we already seen on previous slides, we have not so great picture with some blind spots, with very limited area of color vision and sharp vision, but we have a couple of things that we could do with our vision system to perform better, and the three that we will discuss is first is improve eye, the second is move head, and the last one is move eye. We will go one by one and describe it. And the first way is to improve an eye. An example of such an approach would be a golden eagle, whose color vision is very clear, and it could detect a rabbit in 3.2 kilometers away, and it has 1 million cells on his retina on millimeter squared, compared to only 200,000 for humans. But it comes with some cost and at some price. The price is that size of such an eye would be a very, very big. And it is the same as humans have, but compared to the bird's brain, it's one eye is bigger than the brain, and it is crucial biological restrictions of any bird, because any addition in and improvement in some tissues increasing their weight, and thus it decreases the ability to fly. That's why birds have a relatively small brain, and that's why, for example, humans have a less powerful chews than gorillas and than other monkeys and so on. Yeah, another way is to move head or torso. And here on the right upper picture, you can see recordings of movements for different parts. For example, gaze change, head movement, and right eye movement. And here you can see it for different animals. A is for human, and it is relatively smooth head movement, but we have saccades. This is such relatively small changes in our gaze detection. We will speak more about it in later slides, but on this experiment the gaze in right eye is similar. For example, for a goldfish, it's a relatively smooth head movement as well. But for example, eyes of flies could not move, and they move their body, for example, to move their eyes. And on the bottom picture, you can see that when the bird moves, it have to keep the place of their heads just more and more static. And here is an example of falcon head stabilization and the eye stabilization. We can see that the wind flows such that the bird is not moving relative to Earth, and despite that wings and body moves, the head keeps static. This is how this system works. Very improved eye and very static head position. It enables birds to look on very big distances and estimate everything they need. Mhm. Another way to improve vision system is to move eyes. This is an optical illusion. If you look at one disc, you will think that other discs are moving around, but in fact it is not happening. This is caused by the phenomenon of saccades. By the link you can find other list of optical illusions, not all of them caused by eye movement, but this is caused. And types of eye movements are as following. Here on the right we can see some examples. For example, tremor is a more frequent action with bigger duration, and saccades and microsaccades are more rapid movement. For example, people usually have saccades, and this is the way that we make world around us colorful. As you remember from the previous slide, there is only small area of color vision, and with the help of saccades we move our gaze from this to that, and this is how our brain kind of computes a colorful representation of the world around. And this representation is taken from our vision system, which only have this small area of color vision, but we still percept the world as a colorful thing. Um, and let's look at another example here. This is how the phenomenon of saccades is used. The person sits in chair and looks at the screen, and the mechanism detects gaze detection, gaze direction, and changes those things that person is not looking at the moment. And here you can see the last picture and the first picture and the reaction of the guy who conducted the experiment. You can see that big building disappeared, and the bus disappeared, and the person there on the right disappeared, and many other things, and he didn't notice that. And if this experiment could be conducted to any person who is not prepared for that, because change of the picture happens when our eyes don't look at this direction. That's why we could not notice the change. This is how saccades work. And for example, let's speak about the gap between our vision system, so kind of sensor, and our final world representation in our conscious. How we do that? In this example, our brain tries to minimize imperfection of our vision system, having these saccades, by creating continuous representation of world. And that's why in our consciousness we see this first state for longer. So in our mind we think that the first second will be longer than the second one. This is because a brain changes this state when we didn't see anything. So here is our perception, and this is changed by in this time period, and this is called chronostasis. And let's have some practice on that. Now I will I ask you to change your direction and move your eyes from the screen to anything else, and then I will start the video with clocks, and I will tell you when to move your gaze back, and try to notice how long the first second lasted. Okay, now move your eyes from the screen. I'm starting, and look back. Okay, you could notice that the first seconds that you saw were more durated than the next. And if you didn't feel that, you could repeat it, you could watch the video, or you could experiment with clocks on your wall if you have some. Okay. This is a chronostasis, and it illustrates how our brain, our kind of central computer, tries to make our perception more smooth, and that's how we feel the world continuous. But from the quantum mechanics, for example, we know that world is not continuous. It is more discrete, as in terms of quantums, as and in terms of our vision systems restrictions. And this before in this lecture I have mentioned IATP Vision Lab, and now it is a picture of current state of this laboratory. For example, we have an high-speed eye tracker, color distinction test, and many other stuff that helps us to analyze vision system of a human. And nowadays there are plenty of projects on improving screens, for example, improving video codecs. I will provide some examples in later slides. This is how everything developed, and let's look at some results of this work. And here is an example of eye movement record. Here you see how gazes directions changes for different people. These are points where different people look at. For example, for basketball, and a bit later. And this is an experiment where we measure in more important and less important parts of the video, where people tend to look at and where people tend to not look at. It's made, for example, for marketing research, when we want to improve people's reaction on some videos, when we want to want to make some actions more visible or less visible for our visitors, if it is a film or advertisement or everything. This is always this is usually combined with an EEG signals tracking. This is an electrodes that are placed on the surface of a head, and simultaneous measurements of the gaze and brain activity helps to improve videos and the content. This technology is called neuromarketing, uh, and this part was made at IATP, so we won't look at this more. Now let's look at example of usage in marketing. This is how it's done for a film. So people look at it in a small cinema, and directors look how gaze changes, where people react more, and so on and so forth. In this scene, for example, and more and more and more. So this is a neuromarketing and visual eye tracking for that. Let's move next. Another application of vision systems analysis is, for example, video compression. We could compress films and videos just uniformly with uniformly distributed quality. But another way is to use advanced technique, which consists in packing and in decreasing the quality of those parts of the picture where no one looks at. So we save quality for the most important parts of the video and most important parts of the frame, but drop quality for less important parts. And noticeable result here is that 10 people is enough to estimate the gaze detection for most of the people. This is kind of technologies of future. More than that, nowadays people investigate some models that emulate humans' eye activity. It is used in analysis of key visuals. That's a term from marketing. It is related to the posters of advertisements that you see. So nowadays it's not necessary to have real people who look at your picture or at your video. We could emulate these things. So we will next lectures we will proceed with how it is done, how image processing is done, how it could be applied to some real-world problems in a technical aspect, and so on and so forth. Thank you for the lecture, and I will answer your question at the seminar. Goodbye.